The topic of this screencast is Transcription and Translation, which you will find in Chapter 3 of your textbook. The learning objectives of this screencast are as follows: define a gene, explain how DNA determines structure and function, describe the process of transcription, including initiation, elongation, termination, and the role of RNA polymerase, describe the process of translation, including initiation, elongation, termination, and the roles of transfer RNA and ribosomes. Define a genetic defect, compare germ cells and somatic cells, and explain cell specialization among cells with identical DNA.
So you've probably heard the expression, DNA is the blueprint of life, or something to that effect. You are well aware that you look like your parents because you received your DNA from your parents, and that there is some resemblance in your brothers and sisters, your cousins, grandparents, etc., because you all share a certain percentage of the same DNA. But how does that work? How does DNA determine what you look like, for example? Well, DNA codes for every protein in your body. In fact, a gene is the specific segment of DNA that contains the information for the production of a specific protein or a polypeptide chain. So you can think of DNA as providing the recipe for the making of all of the proteins in your body. In the same way that your mother or your grandmother may have a recipe for making a specific cake or a specific pie or some other cooked item. So DNA provides the recipe for all of the proteins in your body. Remember that over 50% of the organic matter of your body is composed of structural proteins, all of which are made according to your DNA. But not only the structural proteins, but the globular or functional proteins are made according to your DNA. So your protein carriers, cell receptors, hormones, all of those very important proteins which have a large impact on your metabolism are all constructed according to your DNA. Now you may ask the question, well, what about other substances that aren't proteins? How does DNA affect the metabolism, be it the production or breakdown of non-protein organic compounds? Enzymes are proteins, so they're going to be constructed according to your DNA, and those enzymes catalyze reactions that synthesize, break down, and otherwise modify carbohydrates, lipids, as well as other molecules. So DNA determines your appearance, your metabolism, essentially who you are to a certain extent, because your DNA determines all of the proteins produced by all of the cells of your body.
We will now talk about how DNA codes for the production of proteins in a cell. I want to start first by outlining the process, and then we'll get into the details. This figure from your book provides a great outline of the two processes that are involved in the production of protein from DNA. This illustrates the nucleus in a cell. DNA, of course, is found only in the nucleus of the cell, and it never leaves. There, DNA serves as a template for the production of messenger RNA. That process occurs in the nucleus, and it is called transcription. Messenger RNA then leaves the nucleus through a nuclear pore and enters the cytoplasm. There, it attaches to a ribosome, and the messenger RNA provides the code for the production of a polypeptide. And that process, occurring in the cytoplasm at a ribosome, is called translation. So that's the two-step process that is involved in the production of a protein. Let's start with the details of transcription. The first event of RNA transcription is initiation, and it's going to involve an RNA polymerase binding to a specific part of the DNA molecule called a promoter. And there it pries the two complementary strands of DNA away from one another, similar to what helicase did in DNA replication. And with those two strands now separated, transcription can now begin with the formation of a complementary messenger RNA strand. With the RNA polymerase now attached to the DNA molecule, it's going to move along the length of the DNA molecule, and as it does so, it will assemble RNA nucleotides that are complementary to the DNA nucleotides. At first, the RNA nucleotides are hydrogen bound to the DNA nucleotides, forming what's called a DNA-RNA hybrid. In our third and final event of RNA transcription, the RNA polymerase reaches a segment of the DNA which gives it a termination signal, causing it and the messenger RNA to be released. So we've now described the first step in the two-step process of the production of proteins using DNA, and that is transcription. So we've used the DNA to make messenger RNA. Now, technically, what we made was pre-messenger RNA, which has to go through further modification to form the messenger RNA, but I don't hold my students responsible for those details. So for the most part, just understand what we just described as transcription is the production of messenger RNA from DNA. That messenger RNA is then going to move through the nuclear pores into the cytoplasm, where it's going to be used to code for an amino acid sequence or a polypeptide chain at the ribosomes. However, before we move on to describe the details of translation, I do want to mention two other types of RNA in addition to the messenger RNA. DNA is also used to code for ribosomal RNA. What you need to know is those ribosomes that are the sites of protein synthesis, those very important organelles, are a combination of ribosomal RNA and protein. There is also a third type of RNA called transfer RNA, and it's going to play a vital role, as you'll see in just a moment, in bringing amino acids over to the ribosomes for the polymerization of amino acids to form a polypeptide chain. So now let's look at the details of translation. In translation, the language of nucleic acids for messenger RNA is going to be translated into the language of proteins, which are amino acids. The genetic code is the 3 nucleotide base sequence of DNA, which is also referred to as a triplet. This triplet is going to code for a specific sequence of nucleotide bases on the messenger RNA, and that 3-based sequence on mRNA is called a codon. There are 4 kinds of RNA sequences, or nucleotides rather: adenine, cytosine, guanine, and uracil. Therefore, the total possible combinations is 4 to the 3rd power, or 64 possible codons. There are only about 19 or 20 amino acids, so that means that several codons are going to code for the same amino acid. You also have what are called start and stop codons, which don't code for an amino acid, but initiate or terminate translation. This figure from your book illustrates all the different codons and all the different amino acids that they code for, and they're using the 3 letter abbreviations for the different amino acids. You can find a key that'll provide you with the full names of these amino acids, such as phenylalanine, leucine, proline, etc., in your book. So just to be clear, you have a DNA triplet, which is 3 consecutive nucleotides of the DNA molecule. In this case, adenosine, adenosine, tyrosine. That triplet codes for a specific messenger RNA sequence: uracil, uracil, adenosine, because these are complementary to these. This codon on the messenger RNA, if we look over here, UUA, then codes for a specific amino acid, which in this case is leucine. So this is how the DNA provides the genetic code for each amino acid that's going to be formed in the production of a polypeptide chain. Before we get into the details of translation, let me introduce you to our main players. They include the transfer RNA molecule, shown here. Notice that each transfer RNA molecule has an anticodon. That anticodon is going to be complementary to the codon on the messenger RNA molecule. This is the messenger RNA molecule that's being read by this ribosome. And speaking of the ribosome has 2 subunits. It has a small subunit shown here and a large subunit shown here. Notice that the ribosome has 3 sites of attachment for transfer RNA molecules. The A site is for incoming transfer RNA. The P site is for transfer RNA molecules that are holding the amino acids of the growing polypeptide chain. And then the E site, think exit, is the site for outgoing transfer RNA, that is for transfer RNA molecules that are about to detach from the ribosome. Similarly to transcription, translation has 3 events. The first event is initiation, and this is sort of an after the different pieces have fallen into place. But let me show you what exactly has happened here. Well, the first thing that happened was this small subunit attached to the messenger RNA and start running along until it hit a start codon. At that point, the large ribosome RNA attached. And along with it came the initiator transfer RNA, which bears the anticodon for the start codon. And so here we are at the beginning of translation initiation. The next event is elongation. The messenger RNA will be read one codon at a time with one amino acid being added. Notice this was our start codon, and notice that we have added additional amino acids. How is that occurring? Well, here in the A position, we'll have a transfer RNA bringing an amino acid. That transfer RNA has an anticodon to the codon that is in the A position. And so this transfer RNA is going to bind this site in the A position. Once that transfer RNA carrying that amino acid binds to the A position, the amino acid that was attached to the transfer RNA at the P position, it detaches. In other words, the bond between that amino acid and that transfer RNA is broken, and then a peptide bond between that amino acid and the amino acid attached to the transfer RNA in the A position is formed, and that elongates the polypeptide chain. Next, the ribosome is going to continue to move to the right. So the transfer RNA that was in the A position is now in the P position, and the transfer RNA that was in the P position is now in the E position and gets released. And so another transfer RNA is going to come along, attached to the A site with its amino acid. The amino acid attached to the transfer RNA in the P site, that bond is going to be broken, and then it's going to attach to the amino acid of the transfer RNA in the A position. And this is going to happen over and over and over again, and that's going to lead to a longer and longer and longer polypeptide chain until the ribosome reaches a stop codon. At a stop codon, no additional amino acids are added to the polypeptide chain. Instead, a water is added, and that causes the polypeptide chain to detach from the transfer RNA in the P position. And that essentially terminates the production of the polypeptide chain. And that polypeptide chain will then go off and fold and start functioning. Before ending our discussion on translation, I wanted to make it clear that you can have multiple ribosomes forming a protein or polypeptide from a given strand of messenger RNA at the same time. We call these aggregations of ribosomes polyribosomes. So here we have a single strand of messenger RNA, and you can see there are numerous ribosomes attached to it, reading it at different points in time with respect to the completion of the polypeptide chain. So this one has actually finished and detached. This one is at the end. It's just finished the polypeptide and is about to detach. This one is ongoing but has quite a long polypeptide chain, and these are just beginning because they're at the start of the messenger RNA. So this is an array that's moving down this messenger RNA, all forming polypeptide chains from this messenger RNA simultaneously. So it's a very efficient way that the cell can make proteins. And lastly, remember that proteins that are going to be secreted from the cell are formed by ribosomes that are attached to the endoplasmic reticulum. So here we have our messenger RNA that is moving through these ribosomes, which are attached to the rough endoplasmic reticulum. As that as that polypeptide chain grows, it eventually detaches, and then the polypeptide is placed into a vesicle that buds off from the rough endoplasmic reticulum, and then typically is sent to the Golgi apparatus for further processing, and eventually packaged into a secretory vesicle to be secreted outside the cell.
Now that you understand that using DNA to make protein by a cell is a two-step process, you may have the question that I once had, which was, why not make protein directly from DNA? I mean, isn't it inefficient for a cell to first make messenger RNA from DNA and then go from messenger RNA to protein? Why not just go directly from DNA to protein? Well, the answer to that question is simple if you think about, say, the secret recipe for KFC's breading for their chicken, or the formulas for Coca-Cola and Pepsi. Obviously, these companies are not going to give their only original copy out to a production facility, whatever production facility makes their product. They're going to make copies of the original formulas and distribute the copies and keep the original copy tucked away in a safe somewhere where it can't be harmed. Your cell effectively does the same thing. By making messenger RNA, it makes copies of the information from the DNA, which can be used by your ribosomes, the production facilities, if you will, to make the product, the protein. If anything happens to the messenger RNA, you can make more because the original DNA is tucked away and protected in the nucleus. And by the way, the process of making protein is very hard on the messenger RNA, and the messenger RNA is destroyed in the process.
Let's now answer the following question. What is a genetic defect? To have a genetic defect is to have a defective protein or defective production of a protein, and often the two are related. So you can have a defective protein that's produced but actually have normal function, and of course you can have a protein that has been altered, and along with that comes defective function. Let's look at how this could happen. So let's say we have the following DNA triplet here, GAG, that gives us a codon of CUC, and using our table, you can see that CUC, I can find over here, codes for the amino acid leucine. Well, let's say there is a mutation on the DNA, and that A in the triplet is changed to a G. So now we have a triplet of GGG. That gives us a codon of CCC, and using our table, we can see that that codes for the amino acid proline. So this mutation, although small, right, just one nucleotide base, gives us a totally different amino acid in the polypeptide sequence. Now you might think that that one minor alteration won't matter much. However, let's look at what causes sickle cell anemia. Sickle cell anemia is caused by a change in only one of the 146 amino acids that make up hemoglobin. So the hemoglobin molecule, which is a protein found in red blood cells, has 146 amino acids, 146 amino acids long. Sickle cell anemia is caused when the glutamic acid, which is the 6th amino acid, is changed to valine by a simple mutation where the A is changed to T. And this change in just that one amino acid of 146 leads to a situation where when there's a decrease in oxygen in the blood, the red blood cells take on a sickle-like shape. And this causes all kinds of problems. Namely, those sickled cells will get stuck in some parts of the bloodstream and blood vessels, leading to a lack of blood flow to certain organs and pain and sometimes damage and death to tissues and organs, etc. But this just goes to show you how small a potential change in the DNA sequence leads to a change in the RNA sequence, leading to a change in the polypeptide sequence, which can have huge effects.
Cells that contain the same DNA can differ substantially in their functions or physiology, which is reflected in their structure or anatomy. Germ cells are the reproductive cells produced by the testes and ovaries, which are responsible for the production of progeny. Changes or mutations in the DNA of germ cells are heritable, meaning that they are passed down from parents to offspring. Somatic cells are all the other cells of the body. Fibroblasts of the tendons, red blood cells of the blood, fat cells of adipose tissue, macrophages of the white blood cells, to name a few. Alterations in the DNA of somatic cells, as is the case with cancers, are not heritable and will not be passed on from parents to offspring. These cells, illustrated here, all have very different form and function, yet they all share the exact same DNA. So the obvious question is, how can there be so much specialization or diversity among cells of the human body if they all contain the same DNA? The answer is cells specialize by turning genes on and off so that they can form specialized structures according to their function. In other words, different genes or segments of the DNA code for different proteins. Cells all contain the same genes, but they differ in which genes are turned on and which genes are turned off. For example, a neuron activates genes that code for proteins related to neuron tasks and avoids activating genes that code for proteins that would perform skeletal or smooth muscle tasks.
Now let's review the learning objectives of the screencast. Define a gene, explain how DNA determines structure and function, describe the process of transcription, including initiation, elongation, termination, and the role of RNA polymerase. Describe the process of translation, including initiation, elongation, termination, and the roles of transfer RNA and ribosomes. Define genetic defect, compare germ cells and somatic cells, and explain cell specialization among cells with identical DNA.